Hi, my name is Marouen Ben Guebila. I am a postdoctoral fellow at the Department of Biostatistics at Harvard T.H. Chan School of Public Health. I currently work with Dr. John Quackenbush, and today I'm going to present my poster, The Network Zoo, an Integrated Infrastructure for the Development of Gene Regulatory Network Tools and Models. The Network Zoo is an ecosystem of software tools, a database, and a web server that allow to infer gene regulatory networks, to analyze them, to visualize them, to share analytical pipelines with the community. It has 4 different components. The first one is the code base of the software tools that is hosted on GitHub, and which is version controlled in 4 different languages: R, MATLAB, Python, and C. The GitHub repository allows for contributions from community developers to add new features and to add new software tools and to expand on the functionalities of the Network Zoo. The code base is paired with a continuous integration server called the Zookeeper, which allows to regularly test the integrity of the code base to make sure it's maintained and up to date with third-party software tools, but also to make sure that the contributions from community developers are meaningful and maintain the integrity of the code base. The second component is a database of gene regulatory networks called GRAND. This is where we host a set of more than 10,000 gene regulatory networks across human conditions, which can be visualized on the browser and analyzed for using browser tools. Finally, a set of case studies in regulatory genomics is hosted on a Jupyter Hub server, which allows for reproducibility and also allows users who might be interested in learning more about Network Zoo tools to learn and to do their own analysis. These use cases are annotated and curated with additional information about each step of the inference pipeline. And this is motivated by the importance of gene regulation in health and disease. Gene expression, as we all know, is controlled by a set of factors such as microRNA and transcription factors. However, it's not always possible to measure the underlying regulatory network in all contexts. Therefore, we need computational tools to infer gene regulatory networks from gene expression data and to reverse engineer the regulatory network from context-specific data. Typically, we use gene expression from databases such as GTEx, which has gene expression in normal tissues, and TCGA for cancer tissues. This data is context-specific and also publicly available. The Network Zoo itself is a set of tools that allow to infer a bipartite gene regulatory network that links, on one side, regulators such as microRNA and transcription factors to their target genes. We call this the Network Zoo because the animals, the tools that we've been using are called after animals. For network reconstruction, we've been using tools such as panda, lioness, puma, and spider. Once we have the bipartite regulatory network, we can analyze it using tools such as Condor to detect communities in the network, and Alpaca to compare communities in a case and control setting. The next component is a database called GRAND, where we host more than 10,000 gene regulatory networks across human conditions. These networks have been generated in our previous studies to understand gene regulation in normal human tissues, to understand gene regulation between males and females in normal human tissues and also in cancer. GRAND hosts a number of cloud-based analytical tools to visualize the networks, to do functional enrichment analysis, drug repurposing, and differential analysis. Finally, netbooks hosts more than 16 use cases in regulatory genomics as Jupyter notebook. These notebooks are curated and annotated to enable learning and also sharing reproducible pipelines. Netbooks also allows users to create their own analysis pipelines and gives them access to hardware, to disk space, and to RAM space to be able to run these analyses without the need for any hardware or software installed on their computer. Finally, I'd like to thank everyone involved in the Network Zoo community. And if you have any question, please feel free to reach out. Thank you very much.